So yes, we do have a different circumstance in New Zealand. So all of the species that we're working with are taonga, so treasured species. A lot of work, especially around seed collection and seed storage, has been iwi-led. There's a strong social dimension around our taonga species and our interaction with them. And one of particular concern is Maori are quite concerned sometimes about moving germplasm between rohis or regions. So it's not. There's a lot of complexity to our ability to progress forward. That said, I think we've made a fair amount of progress in terms of getting to grips with murder rust in New Zealand. We do have a reasonable store of manuka seed at Plant Food Research at Palmerston North, but that material has been there for some time with permission of iwi. And we do have a lot of information around manuka ecotypes from various programs of research that preceded the incursion. There is some material that Ryan noted was stored in the New Zealand Indigenous Seed Bank. All the iwi groups that I know of are not comfortable with storing their germplasm at that facility because of the way information around that material is handled. The main seed banks we have in the country are more for agricultural pastoral germplasm. So we do have gene banks, but they're not well suited for storing other species. So in particular, Ag Research and Plant Food have got those facilities. There is expertise in the country in terms of a range of the Myrtaceae. So there's, in particular, Karen van der Walt and Gianthi, who spent a lot of time trying to determine ways to maintain Syngeneium, but Gianthi is also responsible for the big manuka store that we have at Plant Food at Palmerston North. And then various other experts, and Emma's here, Peter and Gary from Manaki Whenua, and Peter Delange, a taxonomist from Unitec, who keeps on changing the number of species that we have in the country quite often, which is quite tricky for those of us trying to keep on top of things. So my last count was 28, and now it's 19 species recognised in New Zealand. Basically, all the ecotypes of Canuka have been collapsed into one. We know we've got resistance to Ostrobaccinia in both Manuka and Canuka. We have very hard to find resistance. There might be the occasional plant that's showing some resistance in the other species. The challenge we have in terms of trying to use that resistance that we do have in Manuka and Canuka is that they're actually tissue specific, so it's not just a single gene. A little bit of molecular data that we have so far maps in Manuka the two tissue-specific resistances to two separate chromosomes. So any attempt to make progress through breeding is going to be quite a challenge, should we be allowed to do that. So, but while we got resistance in Manuka and Canuka, at the end of the day, the vast bulk of those two species on the landscape are susceptible. Okay, so it's about 3/4 of the plants that we assessed in the trials here at not here, but in Brisbane with Jeff and Louise were susceptible. The other challenge is, unlike Australia, we have a significant exotic germplasm in the country. About 90% of the plants in New Zealand are introduced. And in fact, basically most of our economy is based on introduced species. that sort of puts a completely different context on how we try to operate. And that includes quite a number. I think someone quoted 200. I certainly see the number at least 150 mutaceous plants have been introduced into New Zealand. Some of them are quite popular. The other challenge we have is we know that it's not just the pandemic pathotype that we need to be concerned with. We know our germplasm is susceptible to the South African multi-host variant, and we also know it can be infected by a Brazilian or Uruguayan, isolate. So breeding for resistance to the pandemic, we don't know whether that resistance would give resistance to these other pathotypes. And as has been noted, for some of the species, traditional seed banking is just simply not available. That didn't work, did it? No. Oh, that's yours, James. Cool. I don't have to talk to that one. Just before, I think probably a couple of things I'd probably like to also just reflect on is our Biosecurity Act makes it quite difficult for us to undertake research. We end up in we've only just managed to get 2 universities added to the list of approved organisations after several years of trying. The Biosecurity Act's there for a purpose, but it also at times can be very, very constraining when you're trying to respond. But that's unfortunately going to be the way it is for a while. I think at that point, I think I might hand over at this point to you, James.
Okay, me again. Last time, I promise. So I think I was asked to speak in this session just to put things in a bit of an ecosystems kind of context, but sort of immediately on the first day of the conference almost, I sort of realised that might not really be necessary. I think we're all on the same page that these things are important, not just because you want to save individual species, but also because you want to save ecosystems. But I'll go through a few things anyway. I mean, this is an analysis that was recently completed using vegetation plot information and a metric of sort of ranking importance based on sort of biomass and also the distinctiveness of the functional role that Myrtaceae fill in New Zealand. And it was the second most important, in inverted commas, family that came up just after the true beeches, which form these completely monodominant stands, so that was unsurprising. But yeah, a really important plant family for the functioning of New Zealand's ecosystems. And then when you break that down a little further, you sort of go a bit more granular and think about the species that we have. That Leptospermae family, Manuka and Kanuka, it's our main sort of cereal species, pioneer species that colonises sort of blank landscapes, whether that's pastures or landslides or so on. And they're really important species for passive restoration, for nurse crops and so on, for the regeneration of native forests in their sort of more climax states. They're both really widespread and important for carbon storage and sequestration, some of the main carbon sinks in New Zealand. They produce a lot of nectar for pollinators, and that's flowed through to them being really important species for the honey industry in New Zealand as well. And the honey industry is especially important agribusiness for Māori. Moving to the next tribe down, Metrosidereae. That's the Metrosidereus species that we have, 12 of them. There's Pahutakawa. This is also a pioneer sort of successional species. It stabilises coastal cliffs. One of the only species that colonise the sterile rocky lava flows soon after volcanic eruptions. Other species of Metrosidereus are canopy dominants, southern and northern rata, also Metrosidereus kermiticensis in Rangitahua, the Kermadec Islands. And also really important for carbon sinks. They produce nectar, just like the Australian Myrtaceae. They're really important for producing food for birds and insects. And some of the species, especially some of the climbing rata, are the only ones that flower during winter. So they produce a food source for insects when others aren't around. And I think something that not many people know, of about 5,500 myrtaceae globally, only about 10 of those are the climbing growth form. It might even be less than 10, of which 6 we have in New Zealand, the climbing Metrosideros. So not only are those climbing Metrosideros important to Māori, like Hone was saying yesterday, they're really important from sort of an evolutionary sense when you're thinking about myrtaceae globally. And those climbing rata occur in about 41% of New Zealand's forest, something like that. That was the back of the envelope calculation that I just came up with, especially in lowland and high rainfall areas. Syzygium maere, it's a wetland tree, one of very few true wetland trees that we have, produces fleshy fruits. It has a patchy distribution now, but it was once way more widespread. It's contracted hugely with the clearing of wetlands for agriculture. And its range is just a shadow of its former self, even as we're talking now. So the last thing we need is myrtle rust to come and sort of deal that final blow. So keeping all that in mind, with all the importance that these species have, how do you sort of prioritise them? I mean, obviously, sort of observed levels of infection, ramarama, pahutakawa, Syzygium maere, or Māori tawake. But there are also other ways, so not exhaustively, but there was the refugia work that we looked at yesterday, so perhaps species that don't have much of their range outside that of the disease. Also other tools like phylogeny, so relatedness between individuals, which we don't have a good handle on yet, and Metrosideros is another example of that. I mean, we've got some idea, so here are our 12 species. The ones up the top here are the freestanding forms, this one too, and then we've got the climbers down the bottom. A lot of work can be done to improve this, and another tree disease we're actually really worried about in New Zealand at the moment is a ceratocystis from Hawaii called rapidohea death. So understanding this phylogeny and the relationships between Metrosideros both in New Zealand and overseas will be really important for anticipating other diseases as well, and it would be nice to, how novel would it be to have a look at this before the disease actually arrives? And just finally, when we're talking about resistance breeding, and again, Heidi talked about it, Richard talked about it. It's about being able to keep and restore healthy ecosystems, not just the species, not just sterile strains and genetic monocultures. Kānuka, for example, that sort of bounces around between 2 species, 10 species, 1 species. One clue from that is that that has a whole range of different forms around the country that have specialised to different environments. So it's about understanding that ecotypic variation and also the genetic variation when you're thinking about how you're going to conserve the species. So work's being done. This map here is looking at shows a phylogeographic kind of the phylogeographic regions for Kānuka in a recent paper. Some progress has been made on looking at the genetic diversity for a bunch of species. This might not be an exclusive list, but they're the ones I'm aware of, so I'd be happy to hear if work's being done on others. But the top 2, Kānuka and Mānuka, that was primarily done partly to work out the difficulties with the taxonomy in terms of Kānuka and for the honey industry for Mānuka. Since myrtle rust arrived, people have been concerned about ramarama and Māori tawake, so there's work being done on that now to characterise the genetic diversity. There might be others too, but we certainly need to get our act together on that one. And then, final point, genetic diversity. We want to restore and promote resistant ecosystems. It's like that photo that Albie put up yesterday of a diverse forest, which is what the end goal should be, both genetically and also by species. That's it from me. Thank you.